10 years between BRIC and Phages. And then for some of you, even longer, like 20 years between MPress or just being with CFWEB or being with Marisa in the classroom when she first came to Montana Tech to do phage discovery. So this is a bittersweet sort of ending, but well, I'll just hold it for now. So there's a but, there's a maybe. So what my talk is about the pedagogical call to action. And so before we begin, like I said, I wanted to say thank you to everyone about that. And thank you for coming today. Our intention is to kind of leave everyone in the room with the motivation, desire, and passion to make sure that programs like Phages can continue. So, you know, this program is funded by the National Institutes of Health and by a lot of moxie between myself and Marisa and all of you to get that funding in the door. But there's opportunities for people to influence local school boards, to influence teachers, to provide more teacher professional development, and then of course influence different philanthropic enterprise that could help things like this continue along into the future. So originally, CFWEB, the Clark Fork Watershed Education Program, which is about to take a name change, by the way. We'll be announcing that in May. So we are clearing the Board of Regents to become a center, a recognized center of the Montana University system. So we will be taking a name change, which will make it easier for people to understand why we work in Baker, Montana when we're the Clark Fork Watershed Education Program. So, but originally we were formed in 2005 to answer the call to help citizens become scientifically literate about what happened in this watershed. And so it was as a result of the lawsuits that the state of Montana citizens filed against British Petroleum ARCO. And the call to action was for us to inform our citizens and specifically K-12 students about the Superfund so that the legacy of Superfund would not continue, that people would understand what happened here and how important it was to protect this area. And then we quickly learned that we needed teachers if we were going to make this science literacy thing happen, because our program, you know, Teal, myself, Chris Doyle before, Dr. Arlene Alvarado, if you remember her, we could see with ourselves about 2,000 kids a year. Pretty proud of that, lots of miles, but it takes a lot more than 2,000 to create a scientifically literate citizenry. So that's where we brought in teachers and started to do a lot more teacher professional development programming. And so these are some photos about why a CFWEP was necessary. So these are historic photos of Butte prior to restoration and remediation. And in the 1890s, the entire landscape of Butte was covered in waste rock and tailings. And fish were absent from Silverbow Creek for over 100 years. And this is a picture of tailings that were impounded in the waterway. That's my childhood Silverbow Creek. We didn't call it Silverbow Creek, we called it something else much more nefarious. And then this is today. This is a picture last Folk Festival. And all of these green hillsides that you see in here were restored landscape that looked like the picture before the black and white. So our students now have the advantage of coming out into Silverbow Creek and being able to look at a restored and healthy ecosystem. It's not completed yet, it's not final, but it's on its way. And so that restoration process is really important in terms of legacy. So this is a quote from Bruce Nelson, Governor Brian Schweitzer's chief of staff in 2007, talking about in terms of the legacy that we leave, this is probably the most important project we will fund, because the legacy of things like the Berkeley Pit, people need to understand how those came about and what we can do about making that a resource instead of a toxic lake. Right? And that's kind of some of the stuff that Dr. Skinner was talking about yesterday, where we're mining rare earth minerals from the mine or from the pit now, from that pit water. So we're turning it back into a resource, and we're also treating that water and being able to release it to Silverbow Creek at aquatic life standard. So something I would have never guessed in my life would come to fruition, but here it is. All right, so our direct impact, this is our brag points. My development director always makes sure that we have to say these things, but this 80,524 students includes all of the students that we've served with BRIC and Phages as well. And 951 teachers, so you are those, some of you. And then we've been in 48 communities. And then these 22 national and regional awards are the awards that our staff and the people that our program have, our CFWEP has personally gained, or the organization has personally gained. When we talk about your awards, well, then it goes way past that. So, because you guys some of you have 22 yourself, so you're amazing. All right, and then here's our outreach where we go across the entire state. And we hope to, with the center creation, to make that even more rich. So if you look at the density colors there, you can tell the Clark Fork is really where we've focused most of our efforts. But then through Phages and Brick, we've been able to expand that to these other counties in Montana. And so we're hoping that that density map gets a lot better, more colors there, darker colors for the rest of the counties as well. All right, so what does a CFWEP have in common with the microbiology program? And it's really the nature of science. So if we think about everything that we're doing outside to study the ecology and understand the restoration of that landscape, it translates into the classroom and into the lab in the nature of science. Like, how do we approach these questions? How do we ask questions? How do we help form an experimental design? How do we help students understand how to do that experimental design? And also, again, back to you all, it's about the teachers that we're helping to inform. And so the nature of science is really where we come together. Marisa and I, when we were first, the very first grant, we're like, okay, how do we fit into each other's lives? And we're like, well, everything has soil, and in microbiology, it happens in the soil too, and that happens outside, and the soil is the foundation for us in terms of the healthy watershed. So we can make this work. We can make it work. And we just also wanted to make sure that we had authentic research experiences for students. So what does it take to create a scientifically literate citizen, as our call to action is requiring us to do? A lot of effort. So a lot of effort, a lot of time. And these pictures are just going to kind of roll through. But our approach is that we can solve big problems, just look at the evidence in our own backyard. And then when we translate that into phages, we can solve big problems with, multiply resistant bacterial infections just by having enough citizens who are engaged in finding those phages and helping us create something that will be a treatment for that. And then just engaging the students where they are as well, and making sure that we connect them to their place and provide them with authentic research experiences that create value and meaning for them, that they're truly connected to what they're doing. It's not just something they're looking at in the textbook that is requiring them to memorize, but rather they have a reason for looking at that information. I love your project, Linda, where your students went out and were looking at the lake water to determine what was going on there, why dogs and people were getting sick and getting rashes and getting infected there. Some of the projects our students on the Montana Tech campus have been running, figuring out stints. Our Goldwater winner this year, she's looking at stints because it was related to her own health and stuff that happened with her health. So that value and meaning invites us to be engaged in the endeavor of science. Sometimes it's hard and frustrating, like Dr. Skinner talked about yesterday, where you're on 15 different iterations, or we can't grow the fish bacteria. How many times do we try to get cold water bacteria going? And it's like, oh my gosh. But over and over and over, right? And then we want to make sure that our students have opportunity to practice doing science. And we talk about science as a practice in the classroom and not as something that you just memorize and learn and then you've checked that off. But rather it's a practice that you have to keep doing it. You have to go out many times. There's lots of times for data collection. We practice it with this context, but then we transition it to another context where we're looking at maybe we're looking at animal ecology this time, or maybe we're looking at how beavers interact on the landscape as compared to just us engineering something. Or again, back to our soil microscopy that we've been talking about, is how does the soil and the biome of the soil interact with our restoration species that we are getting established in this watershed. That's one of my favorite pictures. She's got a tiny mayfly in the tube there, tiny mayfly in the tube. So we have always been rooted in the idea that you have to have our students touching the materials and using the cool equipment. So these are examples of cool equipment, but they're very cheap, right? That's a frame that CFWEP built to do riparian assessment. Your materials that you've been using in your classroom are pretty simple. I mean, they're expensive when we add it up over time for thousands of students, but they're rather simple. The pipetter, when the students touch the pipetter, how amazing is that, right? They love the 7th graders the first time they get that pipetter in their hand, and they're like, oh, this is so cool, and they just want to run it. And then we come out with the automatic pipetter that makes that fun sound, and they're like, oh, can I run that one too? That's touching the things, right? Using the equipment, having the experience of being able to go to the lab with Jim and look in the transmission electron microscope. I mean, I was geeking out. I thought that was the coolest thing ever. Like, I get to run a microscope? You're kidding, right? Like, you guys have done that, right? It's amazing. And then I love the efficacy that we build with our teachers where you create your own materials and you become empowered to bring those to your classroom and show your students that science is accessible. I don't want to say easy, it's accessible, but science is a process and a practice and not something that is only for the people who are the smartest people in the room, okay? And it's rather for all of us. All right, our approach to teacher professional development has always been place-based. So your students scooping their soil is about your place. Them figuring out they could find a phage that was right in their own backyard that could help treat tuberculosis or these other infections was really important to us. We always wanted to make sure that it was relevant and that it was interdisciplinary. We heard from Dr. Skinner yesterday that the interdisciplinary nature of science helps us get better, right? That if we're in our lanes, we don't necessarily come up with the best solutions, but the best solutions come up with multidisciplinary teams. We had a focus on strong science content, so we didn't shy away from the content. We didn't dumb it down or water it down. Rather, we kept it at your level. You know, I was going to say an appropriate level, but it's your level, right? It is where you all are. And I know our first project with BRIC, those of you that engaged in your own research projects hated us at the beginning of those research projects. Absolutely hated us. Students are like, really? Like, yeah, they were revolting. But remember, the teachers were doing things within their schedule, working teacher schedule, and doing the kind of projects that you all have done in the labs, where it's multiple days, lots of hours, lots of effort, and they did it while working and raising families. So kudos to all of them. I think that's part of why they hated us. It's like, this is a lot of work. But they did it, and then they came out with, to quote Sarah, street cred. So they could say to their students, I know what the nature of science is like. I know how to do a research project, and I know how to unveil it. And we felt that that was really important because, again, it creates that efficacy and that ability to mentor you all through those days when it's in the belly of the beast and you don't know, am I going to make it? Is my PhD going to be finished? Is it really going to happen? You know, so they know what that mentoring field can be like and what's needed at those times. And then we've already said this a couple of times, but those authentic experiences. I think that curriculum goes wrong when we read about all the history of science, and then that's what we talk about with research. It's like, well, yeah, it's awesome that Thomas Edison invented the light bulb. That's great, and I love learning about it. It's part of my history that I talk about with my programming in the classroom because it's set up the electrification of the nation. But have I learned science? Maybe, if I try to build my own light bulb, right? Okay, so that's the difference, is doing the science authentically. All right, these are just brag pieces, but I love Linda Ross' quote up here. I've been involved with CFWEP since 2014. I've had opportunities to grow personally and professionally by learning new innovative teaching methods and presenting my work and growth to my colleagues. These programs have been absolutely transformative for me, and I can say with confidence played a huge role in being named 2020 Montana Teacher of the Year. And then she went on to be named a National Teacher of the Year as well. She was a finalist, yes. Yeah, one of three, right? Four. Okay. And then we have Chris is over here as well. who's come to work for us now. She works for CFWEP. I kind of made her. But she says, understanding our place in the world through the lens of watershed science has fundamentally changed who I am as a teacher and as a human being. CFWEP has everything to do with this metamorphosis. The impact of this organization on me and my pedagogy cannot be described as anything other than transformational. So I hope you still feel like that now that she's come to work for us. So this is where I really wanted your administrators to be in the room to hear this. As a group, and this is only the things you've reported, so I know we're missing some because some of you forget to tell me, or Tammy as it were. You've earned 41 local, regional, and national awards, and this is amongst the teacher group. This doesn't include our students and now newly, professionals. 3 of you have completed your PhD programs. 4 have completed master's programs. We had 3 Montana Teachers of the Year, 1 National Teacher of the Year finalist, 2 Presidential Award for Excellence in Math and Science Teaching Education awardees, and we currently have 3 finalists for that same program right now. So please give yourselves a hand for all that you've accomplished. Marisa and I talk about this a lot. We'd like to say that we've had a stake in that or helped in that, but we know that you are what makes this program amazing. Like, you're choosing to come in here and be involved in a program at either 5 years or 10 years of length is incredible. So we understand that we have recruited the creme de la creme, but we hope that you're raising the next cream of the crop. So pretty incredible, if you ask me, in terms of outcomes, right? That said, we did publish. We just got this out in February of 2024. It's about the BRIC project. So if you haven't seen it, you by all means go out there and look at it. We're proud of that work. In this article, we talk about your outcomes of what happened between cohort 1 and 2, and how well the research projects went, and your efficacy and your attitudes towards science research. Teachers are funny, students or former students. Teachers are really funny. They're like, yeah, no, I don't want to be a researcher when I grow up. That was the one thing that they went down on. They're like, nope, I'm really happy being a teacher. Thank you very much. Students tend to go up in that attitude. They tend to say, yeah, I'd like to be a researcher when I grow up. These guys are like, nope, I'm out of here. I'm staying in my program, my degree, in my field. Thank you. But that's out. We are going to work on the next manuscript, which is more about students. This is teacher outcomes, then will be in student outcomes. And I think our plan is for a third would be for about the Phages program. So we'll be writing and be in touch with all of you too. To leave the end of my presentation is that science matters. I know I'm preaching to the choir, right? But science matters for everyone, not just all of us in the room who love it, right? This is a photograph of my kiddo. He now has a giant beard that's down to here, which is funny. But he loves fishing, he loves hunting, he loves the outdoors. And the approach to science and understanding the animal ecology and what's working for animals and where fish are helps him be a better fisher person, right? Helps him be a better hunter because he understands migration routes and where the elk winter and where they need to go for food. COVID-19 taught us that science is critically important for us to understand, right? That when we look at data and we're looking at information that's coming to us through the media, being able to discern is really important. The process that happened with COVID-19 where we got one piece of information and then a few weeks later we get another piece of information was frustrating to people who didn't understand the nature of science. Those of us in the room were like, oh, we just learned more. We know more now. Okay, cool. We figured that out. And we're moving on to the next thing. Now, I was frustrated too. It was like, I don't want to be staying home anymore. But we understood, right? It was easier for us to cope, I think, because we had science background. We had an understanding of the nature. So science matters, and we have to do it early, often, and always. Meaning, we need to have science in school starting from K, pre-K even, and having kids being able to explore and look at things, understand, their world. They're naturally curious at that age. And then it has to be still rooted in the elementary curriculum. And Chris Pavlovich has shown that you can have it as the anchoring phenomenon for your classroom and build all of your other topics around it. Your math can be driven by the science pieces that you're looking at. Your history can be driven by that. Reading in the content area, we always joke that scientists just started reading. And actually, scientists read more pages than any other group of professionals. So we read more than any other group of professionals in our work. So the science literacy and being able to read in the content area is critically important. So why not have that happening in the elementary school all the way along? We tend to not do science in elementary, and then we wait till 7th grade, and then they show up and you guys are expected to do all of the science for them at 7th grade, and they don't have that experience yet. And then we're buying packaged curriculum and trying to get them to roll through that packaged curriculum as if that's creating understanding. When packaged curriculum is really just rote memorization a lot of times. I'm sorry to let out my bias there, but teachers can make packaged curriculum work, but it takes a lot of effort. Okay? Authentic research experiences help students become scientifically literate. So programs like what you've all been through are critically important to our science landscape and to our educational landscape. Guess I'll get off of my soapbox and my preaching, but it is important to us. I think that our communities are better served when we have people who understand the nature of science and are literate. So I leave that with you. I know this is a group of friends and people who are motivated like I am, but I hope that we can kick this off and talk about and start, as we summarize this, where we can be weaving this all together and showing your experiences, our students' experiences, and building that case that programs like this are critically important. So that's all I have. I think I'm transitioning over to Marisa next. Is that right? Yeah. Is this you? Let's see. Oh. Right here. Okay, and then is this one? This one.
Is this the?
Mm-hmm. Yep. There you go.
Good morning. Thank you, Rayelynn,
You're welcome.
reminding us of how important it is what each and every one of these teachers does to reach the students and make a difference for the next generations. I'm supposed to give a talk on the history of phage discovery outreach, but I didn't think it was fair if I made you guys tell your phages journey and I didn't give you mine. So really, I'm just going to talk about my phages journey and throw in a little brick and SEPA while we're at it. Okay, so that's my parents and my siblings and I decades ago. But what was always around in our house was books, National Geographic. My parents didn't believe I could read because I would say the signs, and so my dad opened up the National Geographic, and then when I read it to him, he was like, oh. But my grandfather had a bit of advice for me, and I would just, it keeps me sane. I washed his car. He was this tall by then, about 80 something, and my aunt bought him a Lexus, and he was kicking the bumper. Can you believe it? It's a plastic. This thing is a plastic. Can you believe it? And his car was dirty, and I washed it, and I'm like, oh, sorry, puppy, I missed a spot. He goes, is it better than it was? So that's how I want to judge my activities with education, working with teachers and my colleagues. Did I make a difference in some way? Is it better than it was? So thanks, Poppy. And my mom's parents on the other side, my grandma had to hide her wedding to keep teaching. So that's the shoulders of giants that people, for all the women teachers in this room, you know, thanks Grandma. Yeah, so what I want to point out is that it's that there's joy of discovery, and that one, I should have moved it over a little. It's me looking at some plant, right? That that's what we want to capture in the students. They have it. They're born with it. They're in awe of what they see in nature. And it's really cool if you can make baking soda and vinegar blow up a little container when you're like little too. And it's pretty safe, I think. All right, so then we went to Newbury, Massachusetts, and it's the teachers. Mr. Pike Messenger, I'm still in touch with him today. I write him a letter every year, and he writes a hand card back. And he took us out on his dory in the river, exploring the marshlands. Science team, I wanted to go into biology in ninth grade, and he made me take the introductory physical science class and biology together. He wouldn't let me jump. So I'm grateful to him for everything in science. He's awesome. He cut DNA. He went to a workshop in the summer and cut DNA and run it on a gel like in 1980 something when restriction enzymes were new, right? And so that's what you guys do. You go find out what's the forefront. So thanks, Mr. Messenger. I went to the University of Pittsburgh. My acceptance to Harvard came with and you can work in the cafeteria. Pitt offered me a full academic scholarship. So that was not a hard choice, and I had no regrets. There was an honors college. It was an honors program when I started, and they helped me apply for Rhodes scholarships. I was a finalist one year, and they asked, just walking the application down to the post office, hard copy back then, of course, right? 7 copies, press hard. I realized I was whistling, singing to myself. Like, that's how much weight was on me. And so this is going to come back when I talk about the work I do with students, getting them ready for Goldwater's and other scholarship applications. And I actually was a finalist 2 years in a row. I went to Philadelphia, where Chris just came back from, and I was called, you all are together, these finalists, and they have interviews with a round table of Rhodes Scholars. And in the callback, they said, so, do you see yourself testifying in front of college, in Congress, about science? And I was 20 years old, and I'm like, losing the Rhodes Scholarship in that one quiver. I should have said, well, that's critically important for the future of science. We need our society to understand what science is about and how important it is for our society. But I was 20, and I was just shaking in my boots. So just remember, if you're ever asked that, don't say, So then I went to work, and you'll notice there's really nothing here, because what I learned is that I wanted to be in school. So I went to grad school. What we knew growing, and I ended up so fortunate with Dr. Hatfill as my mentor. You saw him on the video last night, perhaps, just a lifelong mentor. They knew that there was a protein in the bacterium that was required for this phage to stick its DNA into the host. Something in the whole ground up bacteria. That's all they knew. They had an assay and they had the bacteria. So I grew 40 liters of this bacteria and had to spin it down and make bacterial pellets. And I didn't understand that if you spin it for 10 minutes in these little containers, how long it would take to spin 40 liters into a pellet. But I had a big honking pellet by the time I was done at 1 in the morning. Out of that pellet, we purified a protein called mycobacterial integration host factor that corresponded, those fractions corresponded with the reaction assay that we had to make that combined piece of DNA. Cloned the gene in the old days from Cosmid libraries, and then compared that DNA sequence to that of M. tuberculosis and found the protein through sequencing. We N-terminally sequenced and found the DNA and sequenced the DNA. This is when sequencing took days and days and days and days, and found the protein. And we showed that the purified protein bound to the DNA, and it was present, and we found where in the DNA it bound, what bases it covered. I looked this up this morning. I had forgotten what we did. And then we did another study to find out, like, is it essential in the bacterium? When does it show up in the growth curve, late in the growth curve, right? In both Smegmatis and Bacillus calmette-guerin, the BCG vaccine. And we did a fun little thing where you put 2 copies of it in and you mess one up and you see if the cell can lose the not messed up copy, and it could not lose that second good wild type copy, demonstrating that that protein was essential for the bacteria to live. And that's about the last I've thought of it. That was a long time ago. But what do I actually remember? Not that part much until I look at it again this morning. The coffee walks with fellow grad students down the hall, right? And what great mentors we had, because other students had to leave their coats so their advisor didn't know they were taking a walk outside in winter to get a cup of coffee. So what kind of mentor do you want to be, right? Not the kind that the student has to hide that they're taking a break. Jeez. So anyway, just stuffed these in this morning, forgot to turn it. That's my mom with me at the Olympics, Dr. Hatfull, amazing mentor. American Gladiators was something I did, nobody told it. This is the secret coming out of my closet. There's a 1990s something version of American Gladiators, for real. I won. $2,000 when you're a graduate student is like half a laptop. And I spent so many hours in my judo club basement, I can't even tell you, with the weight room, and eating pizza in between sets. I had the guts of steel. And then I went to a postdoc at Washington State, where Leanna was born, and came back to Pittsburgh to work with Graham again, sequencing phage genomes. And that's where Kyle was born. So it is really when I came back, right? This is when the phage discovery outreach was born. So that's now my talk starts. So in the early days, Bill Brucker came into the lab. He was a student from Brown, football player. HHMI provided him a fellowship to come in for the summer, and he discovered some phages, corn dog actually, and he showed it could work, right? And so Bill Jacobs is a famous scientist, and his twin sister lived near Pittsburgh, and he said, "Go pay Graham a visit." Well, she was a teacher, and she recruited 2 students to come into tech, and you can see these big printouts of genomes on the wall, right? So she got Joe and Jake into the lab, okay? And again, they showed high school students could discover phages, right? And then the idea was they don't have to know anything. They don't have to know anything to start. Just here, you push this button, lift this up. We talked about that yesterday. So ideally, student research projects, if you want to think big scale, national scale, have to be technically feasible. It's frustrating when stuff doesn't work, and if you only get 1 or 2 shots at it, well, what a crappy project. So that's why I thank Dr. Hatfull for my grad project. There was a protein in those bacteria. We knew it. It would work. We could purify it. I wasn't going to win a Nobel Prize, but I was going to get a product and have a degree, right? And it taught me a lot. Scientifically meaningful, motivating, place-based. What is the motivation for doing this? You contribute to, okay, interruption. Welcome, Chris. We're so happy you're here. Yay! The students actually, it's theirs, their sample. That we can accommodate a large number of students. Stay tuned for big numbers, right? Like you guys have brought thousands of students into this. And peer mentoring and near peer mentoring, which we'll talk a little bit about. They can help each other. It's so cool to see the students go, no, hold that there, no, push that, or help. One student holds the tube and the other one does the syringe, right? And so it's easy. 1, collect sample. 2, filter. 3, infect. 4, confirm. And the motivating part, students get to name the phages. Their name is on PhagesDB as the co-discoverers. And believe it or not, those students, citizen scientists, can help us answer what does biology actually want to know? What's on our planet? How did it get to be that way? I mean, that is fundamental biology. Of course, there's the biological and medical relevance of antibiotic-resistant bacteria. That's very motivating for students. Maybe I could save somebody's life with this phage. But it's easy. Isolate phages and name them. Then it comes back to the students at Tech. or amazing high school students who work in their high school labs, or teachers who purify and prepare high titer stocks, isolate the DNA. We send it out for sequencing. It took 4 years to sequence the first phage. PhD, whole paper. Now they can do them in a day, $200. Annotate the genomes. Lots of students have done that. There's a lot of programs now that make it easier than the pencil and paper that I did when I first started as a postdoc, and compare those genomes to others. Our first 100 high school students through the first stages, we went in the evenings. This was a IB, International Baccalaureate. And then since then, it's kind of gone viral. Over 100 colleges from Dr. Hatfield's C-Phages program, over 25,000 phages, he told you last night. I think it's about 4,800 complete phage DNA sequences, and papers describing phages and the outcomes for students, right, for these college students working on this. A key point I wanted to bring back is undergraduate researchers as mentors. This is Kostya. He was from Albania, and he came to this classroom with me in 2000, what did I say, 3 it was, or 2? It was a long time ago. But we compared the genomes, rather than one paper for one phage, 14 phages. We got the cover. We got their first centerfold with the maps of the genomes. So that paper, it makes a difference to write up your stuff and tell people about it. And so then that was at Pitt. I don't know why. I do know why, but we came to Montana, and this was my job talk, 2005. Bacteriophage isolation and genomics, vessels for student voyages of exploration. So I tried to sell them on the idea that phages were the perfect topic for undergraduate students. Really advanced PowerPoint skills. But Sandy Wardell had been in Pittsburgh that summer, and she invited me to Helena High, fall of 2005, driving up I-15 in the dark. I didn't see an entire car the entire way, and from somebody from Pittsburgh, I thought I was in Deliverance. I'm not kidding. I was like, where are the other cars? We didn't have the, iPhone GPS, so I'm just driving in the dark at like 5:30 in the morning going, what the heck? But it was awesome, and her students were awesome, and here's Andrew, Dr. Ramstead, undergraduate mentor, came with me for one of those visits, and that was 2005. And then I met Rayelynn and got involved with CFWEP over the years. And we decided to write for an NIH SEPA award, now NIH Science Education Partnership Award. People had told me this is a perfect fit for phage discovery, but I didn't know a lot of teachers other than the ones that Sandy had introduced me to, some of whom are sitting around, and Megan, and I met Kate through CFWEP, and I was actually at Capitol High with very little memory of a long time ago, just on a single couple of visits. But now over 13,000 students and 50 teachers, and it's because of the partnership with CFWEP and these NIH SEPA awards. We wrote it in 2012. We got an amazing score from the reviewers, and then sequestration and the government shut down. So we were just crushed. And then we were sitting at lunch, and we're like, well, we should email he's like, didn't you get my email? Well, what's her title? The program director let us know that he had sent us our award letter, but we changed the dean of the graduate school and vice chancellor for research, and the email was never updated, so we didn't know we had an award. And then we're like, yeah, you got an award. So we flew to DC and went to our first SciEd conference like within a couple weeks, and then got the letters out to invite teachers to join us. And there were 28 teachers from Libby to Baker, and we just had an amazing time. Some highlights that just like blew us away. This was an original plate, thousands of plaques from the Boulder River, Steve McCauley's class, and then Sentinel High. There were 2 different phages on this plate, Prickles and Novi 14. Students all over the place. I had a sabbatical. They worked on Gauge AP that was discovered by a 7th grade student in Megan's class, and then purified, annotated by high school students at Helena High. H. Savage. Was it Helio Savage, Grace? Yeah. Renamed, so Grace is here. Worked with Hannah and did the entire map and won all kinds of awards. I don't have to show you this. Rayelynn showed you that we just finally published some of the teacher findings from that, and then got another 5-year award, 6 teacher leaders and their teacher partners, a partnership, and Chris is in the building. He drove over 30,000 miles between these projects, so into your classrooms. Amazing people. And now these are our teachers, as mentioned yesterday. Over 130 phages, 21 are sequenced and annotated in GenBank. During this time period, Dr. Hatfull and people at Pitt with their 20,000 phages plus in their archives have successfully used them to save lives. Isabel was a 15-year-old cystic fibrosis patient. More recently, they out of 20 individual case study trials, 11 of those people survived, over 50% survival with people who were terminally ill with superbug infections. So yay, I don't have to say maybe someday anymore. I can say the phages discovered in this program have saved lives. And then to come back to full circle with Pitt nominating me for awards, in 2005, former Chancellor Gilmore at Montana Tech asked me to start up a National Student Awards Committee. And since then, we have 11 Goldwater Scholars. Tech has 12 total. There was one before we got there. Just one was announced the end of March. And it was the last 2 years in a row, they've been transfer students who came to us from community college. Found out they can do research. They like doing research. And they used to have honorable mentions. We also have 5 honorable mentions. 3 finalists interviewed for Rhodes, so we do practice interviews with them. And the one that was just mentioned, Bailey Phillips, just defended her master's, and she's going to go to the University of Washington to pursue a PhD in biomedical material sciences. I'm not sure exactly the degree title, to work on vascular stents, because that's what's important to her. And athletes, and Bailey just found out this week that she won an NSF graduate research fellowship that's going to cover all her expenses and give her a livable salary in Seattle. So, and so Phages was supposed to drive these teachers here sitting in this room and partner with them and get them to independent delivery of phage discovery in their classrooms. And what we found out, shockingly, was that COVID actually accelerated this, because we couldn't come in person. We couldn't, and they learned to deliver it themselves. I did some horrible recorded PowerPoint slides. I learned how to use this program a little bit, how to use it, called Zoom. And Zoom's pretty good when you're recovering from back surgery, if you want to see your students, by the way. It's pretty good. But it might have accelerated it. Like, we were kind of, okay, that's something, so some silver lining, I don't know. One of the things we're really proud of is that we developed this pipeline program. And the idea is we want to get these students into a very open pipeline to study science, to become biomedical researchers, or whatever careers they take. You'll see today and tomorrow students taking all these different careers. And I'm sorry I don't have more time to go through each and every one of these students, but they were awesome, and every year they keep coming back, and former students become the mentors. And if I missed anybody, I'm sorry, but a lot of these students are sitting here and going to give a little talk. A lot of the students with advanced degrees were here from my early days at Montana Tech. The CPhages database has grown. This was, as of last week, over 100 colleges, this many phages. We have 21 sequenced and annotated phages from Montana Tech's work, and students are authors on those. Some teachers are authors on a couple of them. So it's really thrilling. And just notice it's not mycobacterium only anymore. They have all these other hosts, and Casey, who's going to give a talk, I'm not sure when, tomorrow, is annotating. We didn't discover it, but she's annotating a streptomyces phage that has like 230 some genes in it. So good for her for taking on the big challenges. K-20 students can continue to do real and meaningful research. And I think the Phages project, it's evidenced that teachers can provide real, meaningful research to those students. And they can do it independently if you give them the tools, an incubator and an autoclave, and a bunch of colleagues who have their backs. If they don't need SMEG and they have SMEG, then they can share it because it grows. Good job, bacteria. Good job, teacher colleagues. And I'm so excited Dr. Driver's here. And if you remember from one of those early slides, there was a student in a microscope. Well, that was in Missoula, and that microscope doesn't work anymore. And I was like, well, what the heck, let's write a proposal. We have 6 weeks. You're getting married in that 6 weeks, and it's winter break. Why not? And I thought I had about zero chance of getting this. And I was a bitter person. And I don't want you to be bitter when you take a chance, because I got a call 6 months later, right before the wedding party. Didn't you get my email? Check your junk mail. We need this by tomorrow, this information. Change it to a 3-year grant, not a 5-year grant. Same amount of money, only writing 3 reports instead of 5? Yes, ma'am. But here is our new scanning, STEM, transmission electron microscope. It now has advanced EDS capabilities on it, thanks to Dr. Skinner and the ARL funding. And Dr. Driver's here, and he does not work at the University of Montana anymore, because we've lured him with a brand new microscope. I will not use the word poached. We lured him here because he loves to work with teachers and students, and that's why I got this grant, because I wrote about what we will do with this microscope with you and your students. I was advised to take a lot of stuff out of my grant, and I held my ground, and here we go. $1 million microscope. It's amazing, and we just had advanced training on it with the Hitachi people, and Thursday we're going to have advanced training with that EDS from Oxford people, and that machine now can do it can jump horses over barriers. I want to make sure I mention this, all these students who worked very closely with me at the sabbatical, all the BRIC teachers, the Phages teachers. What a group. You're so inspiring. Sandy Wardell always inspired me, because every summer she was off learning something new. And that's what you guys do. And that's what makes a difference for your students, because they're not hearing about what was in the book 11 years ago when it was actually written. And that's what Mr. Messenger did. So mentors and heroes, my parents, they read to me, They let me play in the dirt and climb a tree. And when I got stuck and scared at the top, my dad talked me down. He didn't come get me. Right? You can do it. You can figure it out for yourself. With help and support in the safety net. He would have caught me if I fell. My coaches, still one of my best friends, Colleen Murphy, Nick Zafuto, Dr. Hatfull, was my PhD advisor, and I went back as a postdoc to get the outreach, and that's when the outreach was born, working with him. My colleagues, it's just, we can't do this. One person cannot do 13,000 student visits, right? And all my students over the years, I had a couple pictures up there, but I can't get them all on the one slide. So thanks for coming back and talking. Pioneers in science, athletics, and life. Dr. Hartline is a staunch ally and supporter of women in science, and that matters at the administrative level, right? At the national level, to get these young women into Goldwater Scholars, into research labs, into whatever they want to do. It's awesome. And I tried to get all the names on here. I sure hope I did. Some old pictures. Some of these people are sitting in the front row. And it's been a great time. And what matters to me is what they go off and do. I get emails that make my day. Oh, my God. Grad school. It was actually med school. Everybody was struggling. And I had to explain molecular biology to them. And one of them told me, it's not any harder than your class, it's just more volume. And so I'm guilty of fire hosing students sometimes. I admit it.
No way.
Says the front row. But really, it's my family. Leanna was born in '02, and I only own one dress, but Kyle was born in 2002. COVID wrecks lives, guys. He was at Boise State locked in his room for 3 months and never ever wants to see a college campus again. So life happens. We adapt, we adjust, and all's going to be great because of the good people that they're going to be. They are. And Mike joined us in 2011. Talk about good people. Took on these guys when they were this big. And Saturday, he was standing at our neighbor's fence, and a guy passed out driving up the street, going about 50, plowed into the fence, blew it up. He yelled, "Watch out," to me. The neighbor was there. The fence is gone. The neighbor dove, and the wood hit both of them. Crashed through a telephone pole and into my FJ. That lets you know what matters, right? And even the driver was okay because of airbags. So yay, science and technology. And boo passing out. And Cher's a rock star girl. Just treat them nice and they'll become nice. I have no idea what time it is. How are we doing?
9:26.
Oh, rock on. I think this is Anka Segal and Forrest Rauer's sculpture in San Diego. That's Phages art. Any questions? None? Really? Well, I love you all. Thank you.
This is so much fun. It's like the memory lane things. We were talking about that in the